So I was worried that I would repeat a lot of what other people say, so I decided to talk about something that is a little different. So let's look at the CML landscape in 22 in high-income countries. I think there's a perception of a solved problem. There's increasing prevalence, prospect of indefinite TKI therapy for still the majority of patients. Therapy is moving from academic settings to community. Ongoing struggle with high drug prices, not everywhere, of course, but certainly in some countries. Important questions moving outside the expertise of clinicians and leukemia biologists. Reduced research funding and less interest to dedicate a career to CML. So, based on that landscape, I think it's really time to think a little bit, not just about the biology, but also about how we address that in terms of utilizing our resources. I don't have to tell you this. This looks excellent. 10-year survival of the IRIS study. And I think there is a notion that in resource-rich countries with very good medical care systems, the survival of CML patients approaches that of the general population. This is a study from Sweden that quite obviously shows that. Do I have a pointer here, by the way? Huh? No. Okay. Now, another issue, I think, is that CML has, I think, had the advantage of being a paradigm disease, and a lot of research expenses went into CML because it was one of the first diseases that really became open to being studied because we knew the molecular abnormalities. However, other cancers have caught up, and if you just do a quick PubMed search and you compare, you search for CML in the title versus cancer in the title, and then you do the percentage of CML versus cancer, you see that this peaked in the 90s or early 90s, and then kept at a pretty high level for about 2 decades, but over the last decades, it's declining. And that just means that the resources that are being spent by societies on cancer research are less likely to end up with CML. And the same, pretty much the same or similar data you see if you compare CML to other types of hematologic malignancies. So I think it's something we need to bear in mind if we want to keep pushing and keep the CML field alive and vibrant. Along the same lines, research funding, NIH or NSF, that's the National Science Foundation. So I'm just comparing a CML, AML, and pancreatic cancer. And the data speak for themselves. So this is really what we're up against, that federal funding agencies make it harder to obtain peer-reviewed funding for CML-based research, maybe for some of the reasons that I initially mentioned. At the same time, I think, at least I frequently just make the mistake of looking at things way too much from the perspective of a resource-rich country. But if you look at other countries such as India, and we have fortunately a colleague from India here in the audience, it looks sort of scary because the population is growing. There's probably even the incidence growing, and as such, the number of patients who are in the CML world is increasing. And of course, that really begs the question of how resources are allocated. It is quite clear, and maybe the question that I posed to Tim was not a really good introduction to that, but it's quite clear that if you let the disease go for a significant period of time to a stage of advanced phase CML, it's way more difficult to control. So the earlier you hit it, the better the outcomes. So in high-income countries, we are fortunate to see a lot of patients who get diagnosed because they have an abnormal blood count. This is not the case in less fortunate countries. And so if you think about where would you put the money to have the maximum impact, then I think, unfortunately, and to my own dismay, the molecular technologies that are really attractive and fascinating are probably not going to get us the maximum impact we could get for that amount of money. This is from a manuscript that Cherry Redditch and Dr. Malhotra authored a few years ago, and I think Cherry made up this equation there, so there's probably no way to calculate that. But I think it makes the point. So we really have to position ourselves a little bit in terms of where we allocate our resources. So talking about developing countries, well, I mean, we are kind of in a developing country here, because if you look at the outcomes of CML patients in the United States, it is quite clear that while overall outcomes are much better than they were 20 or 25 years ago, this benefit is not spread equally across the population. So if you are older, if you are Black, if you are socially disadvantaged, if you are on Medicaid, or if you have no insurance, your outcome is going to be worse than that of the rest of the population. And the differences are actually, not all of these differences are significant, okay? But I think it is a warning sign that despite the high healthcare spending and despite access to the latest technology, we have failed to solve the problem for a significant proportion of the population. This is, by the way, typically these are SEER data. SEER is surveillance I can never remember that, but it's basically a cancer registry that derives up to 20, or is derived from up to 20% of the population. So along the same lines here, this is published a few years ago, and now we look at CML survival in the US according to what it should be and what it is indeed. And again, you see these differences. And lastly, I think Bob Gale published that in Leukemia a couple of years ago. The red curve in the right lower graph is the actuarial survival, and you see that there is still a significant difference to a population-controlled survival or survival rate. So, given these limited resources that we're looking at, is it time to reconsider some of our approaches, or should we just basically stay the course and continue? Now, I have a few questions that just strike me sometimes. And this is not to really badmouth any of these papers here, but I think what we sometimes do is we just research the same thing all over again, time and again. So every center does it, and this is looking in this case at the BCR-ABL1 transcript types and their impact on survival, and it's not complete, so there are multiple studies. I think at some point we just have to say, we know that. And the resources should probably not go into another study to look into that. Number two, sometimes I think we also get a little bit too obsessed with details, and I take blame here. So this is a paper published about 15 years ago, where we looked at the transforming ability of BCR-ABL kinase domain mutants, and you see some differences here. But honestly, that's probably $500,000 spending. I don't think it really had a major impact. So, of course, that is hindsight wisdom, but in a research source stretched situation, I think we have to think carefully about what projects we embark on and where there is potentially high impact to have in terms of patient outcomes. So it's somewhat like a Mandelbrot system. Number three, is everything knowable worth knowing? This is a study that looked at combination treatment with propofol and TKIs, and found that propofol and TKIs are synergistic, and so the conclusion is that propofol has potential to be repurposed for CML treatment. Well, I think I would say likely not, because as you know, Michael Jackson tried that, and it had a really bad outcome. So do we have to do this kind of study, right? I mean, what is the end game here? What are we learning from that? So how do we deploy limited resources to achieve maximum impact? And this is where the title of my talk comes from. Is it free treatment for as many patients as possible? Free treatment meaning free medical care, effective medical care for as many patients as possible? Or do we say freedom from treatment for as many patients as possible as the really ultimate solution to the problem? And somewhere, of course, the truth is somewhere in the middle. I made this myself, you won't believe it. So treatment-free remission. I have a few questions. Will we ever be able to predict TFR success? Well, I would argue, do we need to predict that? Because if we really think that everybody should have a chance to do that, could we just say try as many times as you want? Do we have to identify these patients? What is the level of certainty that we would have to have clinically to tell a patient, you'll never be a candidate, or you'll be a candidate no matter what? Is there any such level of certainty? I don't know. Do we use the right disease model to understand persistent leukemia? Is TKI plus X the answer to the problem? And lastly, are we translating all the beautiful preclinical data we have? So I probably can't operate this thing, but okay, associations with TFR. You know, I don't have to repeat that here, but again, there is plenty of data, and I think what we haven't arrived at is a test that is 100% sensitive and 100% specific, which would be ideal, but it's probably impossible. And if I could operate the system here, you would see these two pendula. So one is basically a single pendulum, the other one is this double pendulum. The difference between the two pendula is each time just 0.01%. But what you'll see is that this single pendulum will swing in synchrony for quite a while, whereas the pendulum on the other side is turning into a chaotic system within a relatively short period of time. Now, if you think how much time goes between diagnosing a CML patient and trying TFR, you could think about a lot of stochastic factors that will ultimately contribute to the outcome. And so maybe it's just not possible to do that with the degree of certainty that we would like to have for what we have to consider a low-risk intervention, TFR attempt. If the right was going to work, I would show you a famous video from the BBC that is a weatherman of the BBC called Michael Fish, who is known to predict or to announce in 1987, here's the BBC weather report. You know, a woman called the BBC today saying a hurricane was underway. And of course, this is not true. There will be some rain here in southern England. And this was this remarkable hurricane of 1987 that really wreaked havoc in the south of England. So that is just to say, prediction models, even the best ones, can never really be 100% precise. So how many TFR attempt until it's time to give up? Maybe unlimited, right? There's probably no limit to the number of times you can try that. So is that something we should just consider in every patient? And is there a worry that maybe in the real world the TFR curves are not as nicely plateauing as they are in clinical trials? So in other words, will we just see a lot of late recurrences at some point? I think the jury on that is out. And then lastly, all this beautiful research work, the Holy Grail, finding the weak spot in CML stem cells. As you know, we have generated many beautiful in preclinical data sets to identify various critical factors that are required for CML stem cells to survive. Unfortunately, very few of these targets were actually tested in clinical trials. So despite this information, we haven't gone the next step. And there is, of course, many reasons for that, but it is sometimes you start thinking, well, how many more of these data sets do we need? Because we are not really putting that to the test of the real world. And so these are, in this case, it's just HDACs and hedgehog inhibitors have been tested in clinical trials. And this is a continuation of the list. And again, only these 3 have been tested in clinical trials. So if you do the math, it doesn't look very good. 24 targets, and only 2 of these targets reported out on a clinical trial. So we have kind of now convincing information to say, no, it doesn't work. We don't have to touch that again. So I think for us as a community, I think we just don't look terribly good, if you're honest. So, of course, there's a number of obstacles to explain that. I think one really important one is that we have still a disjunction between the world of basic biology and the world of clinical research. For a basic scientist, it's about the next grant and a high-risk, high-impact publication. For a clinician, it's the ability to run clinical trials in an complicated system of clinical care, and everybody who knows who runs clinical trials in this country at least knows that. So I think progress here will actually not require more biology. It will clearly require more collaboration and just maybe a fundamental rethinking of how we do clinical research. So I think really this is probably up for disruptive innovation. And then humans are not mice. So a lot of the most convincing data is based on very strong and very rigorous mouse models, but they just sometimes don't pan out in humans. And this is one example here. This is about a gene called LOX5, which is a lipoxygenase. So in these left data, it was shown that this is really important for the survival of CML, of primitive CML cells and full chemogenesis in mouse models. It turns out if you look in humans, the gene is actually not expressed in the most critical primitive cells. And if you inhibit it with a small molecule, siloitron, it doesn't do much. And as a result, it's not surprising that the clinical trial didn't show anything. So I think on us, it will be to say, hey, before you get to publish in a high-impact journal, you really got to show us something that has human cells to make that convincing. And then lastly, well, this is just messy as well. So we like these clear-cut pathways, linear pathways, but biology is different because I guess you could say otherwise life wouldn't be possible if it wasn't messy. And so the situation is such that there are mutations, there's absorption of drug, there is compliance, there is all this stuff that impacts responses, and it's very difficult to tease out. Clearly, I think now with technology available, the next step is to go to single cells, because that is a level of resolution that wasn't possible so far. Well, this is just to illustrate that this is a collaboration that is hopefully at some point going to get published with Brian and Cherry Redditch, looking at RNA sequencing from patients with clinical BCR-ABL dependent or independent resistance. And just not to go into too much detail, but what you can do is essentially you can group these genes into co-regulated brackets, and then you say a type of resistance, what kind of brackets is activated in that type of resistance. And so you can use that as a way of classifying patients according to the activation or inactivation of certain brackets. Now, I think this is kind of old data already because it's still bulk sequencing. I think the future is really to go into single cells. This is just an example. Patient with CML, T59 mutant, not responding to ponatinib at the highest dose. And so we are trying to use this approach to understand what is wrong or what is special with the primitive cells in these patients to maybe understand what specifically to use to target these cells. Now, I think if one wanted to end on a positive note, this is really what one would like to do, because technology in terms of inhibitors has moved forward really a giant step. And so I think until now we really couldn't clarify the important question, how far can we get by just inhibiting BCR-ABL really, really well? And we had some hints from the EPIC trial, which of course was abandoned for cardiovascular toxicity. But there is some data available, and they are admittedly thin because the trial closed early. But if you look at the responses here and compare them with imatinib and nilotinib responses on the NSND study, you clearly see that by and large you see a doubling of the responses. So this is possible if you really hit BCR-ABL very hard. You get to this response levels, and I think this is maybe a little, even a little better than ABL001 or asciminib. But this is, of course, an inhibitor that is not ponatinib and is extremely specific. And for those of you who are not familiar with these diagrams, this is basically a cell line panel, and you graph the IC50 values versus the genotype. And so if you have something like asciminib in the very first row, where all the significant hits in red are associated with a BCR-ABL positive genotype, and not much happens on the right side, you know you have something that is very, very specific and doesn't have, presumably, at least some of the problems that the other inhibitors have. And the last, I think PROTACs are coming along. So PROTAC, this is for protein degrading chimera. PROTACs are in clinical trials for indications mostly in the field of solid tumors, but people have tried or have started to make ABL-directed PROTACs as well. This is work from Brian's lab, and I wanted to make two comments here. One is there are clearly functions of BCR-ABL that are not kinase dependent, and we had known about these for quite a while. For instance, Dr. Bhatia published on that the migration defects and the adherence defects are not really requiring active BCR-ABL. And we found a few years ago that mislocalization of P27 is also not dependent on kinase activity, but is actively contributing to reduction of apoptosis. And if you look at the right here, this is basically a comparison of cell lines grown in the presence or absence of serum. And then the second variable is the ABL PROTAC versus an ABL inhibitor. So basically you have either degradation or you have just inhibition. And if you look at the third line from above, in the presence of serum, in the upper graph, there are these two empty, lanes, and this is SHIC. This is a target of BCR-ABL that is dephosphorylated, but only with a PROTAC. The inhibitor doesn't do it. So there are scaffolding functions of BCR-ABL that are not amenable to inhibiting BCR-ABL. And so you could argue maybe having a PROTAC that gets rid of BCR-ABL would be able to do something that a TKI is not able to do. Okay. So to me, the next frontier is improving access to care, actually doing the clinical trials, testing the limit of BCR-ABL inhibition, charting the CML ecosystem at the single cell level, and lastly, maintaining the CML research community. And I've been blessed to work with a number of absolutely great people, and so let's just keep on pushing. Thank you.
Right. Okay, questions for Mike. That was a great, very thought-provoking talk. Jorge.
Thank you, Mike. A great presentation, because you brought up a number of very good issues, and more than a question, it's a comment. Part of the problem that we face when you pointed out all the research that's done that probably advances very little and I'll bring up, for example, the one that you did, which I actually like a lot, about the potential of each mutant to be, have an advantage or not. Those things get us grants. The practical answers to practical questions, they don't get us grants, and support from, organizations and others to investigate some very practical aspects. And that's one of the problems, that I think that the money, the support money, goes to very specific purposes, not necessarily the general questions that apply to more of the population in the world, but even in the US, because, I mean, the reality is that the patients that we study, that get into the studies, are very selected patients, not the patients that everybody else sees around the country.
Well, I just have to agree with that. So, good comment.
Thank you. I just want to add to what you've said. I wrote in my book, you said the funding from NIH has decreased and they have funding more for pancreatic cancer. And I just want to say, I think CML has become the success that you had with imatinib created. So I would say CML is his own worst enemy, in the sense that you created this, you made it a chronic disease, in the sense that you give in tablets, and now they see it as not as unmet need, as you say, for pancreatic cancer, that within 6 months the patient is gone. And I think we just need to get the attention of the people who have the money now to say that even though pancreatic cancer, they die in 6 months, people are still dying with CML. Thank you.
Comment. Okay. Yeah. Pepe. Yeah. Thanks.
Thank you very much, Michael, for this very beautiful presentation. I would like to say, coming to one of the first slides in which you were mentioning the differences that we are seeing in treating different populations, I would say, within the same country, as in the case of this USA, or around the world. So I think that, of course, we are generally speaking, even in this meeting, of trials that cannot be applied at the end of the day in most of the countries of the world, because of the price, because of several other questions. And then also the population in which we are performing this trial is different, because in many countries, CML patients have access to assistance when they are high risk already, with a high, most of them indeed are already high risk for the burden of the disease and other reasons. So I think, don't you think that it's time to think of more specific trials to be run in specific countries, knowing better, I would say, the epidemiological situation of the disease in this country, and to try to find accessible solutions to cure these patients. Otherwise, we are just, for the moment, we have imatinib, which is beautiful, I would say, but in the talking about TFR and so on, and to complete the defeat of the disease, I think that we need a wider point of view. So,
I could answer the question, but I would really want to ask Hemat, Dr. Malhotra, to take it, because I'm sure he's going to speak to that very competently.
So I think appreciate Michael, highlighting the problems which half of the world and maybe 60% of the CML patients in the world, they face. There is no question that we diagnose the patients in much later in the chronic phase, and where the genome is a lot more unstable, and possibly there are more genetic mutations leading to less optimal response as compared to the developed country counterparts. And there's no doubt in my mind that a different strategy, both for controlling the leukemia, as Tim was saying, and also for keeping it controlled for a longer period of time before you attempt TFR, are needed in like half of the world. So the guidelines which ELN lays down or the NCCN lays down and which most of us, my colleagues and myself included, we blindly follow, that needs to change. And when will that change? When we have our own data to be able to tell you. And that is happening. It's not happening as rapidly I would like it to happen, but it is certainly happening. And I don't think the biology of the disease is different, but just that the starting burden of the disease is very, very different. So different strategy, as what is written down, a different strategy to that is really truly needed.
On that point, Hemant, are you able to contribute to the TFR Alliance? Because I think looking at different countries and the variables would be very valuable for that particular
100% agree, Tim.
And it'd be great to have more contributors from India, because I think that's a really important point. And it'd be fascinating to look at a cohort of Indian patients. I mean, Narani's work suggests that about 30% of patients at diagnosis have additional genomic events, and 70% have nothing else that we can find. How would that be in the Indian population? It'd be wonderful to look at that, to see. I mean, I suspect you might see a lot more with the genomic lesions, but it'd be a wonderful opportunity to do that. Other questions from Mike? We still have, he's going in a sec, so it's your last chance. Francois.
Well, thank you very much, Mike, for this very nice presentation. Just a general question linked to what we have said before. In fact, as you explained very well, it's a kind of stochastic disease because, it's an event appear in the stem cells, but the frequency of the event is very reproducible from one country to another country, as compared to the acute promyelocytic leukemia. The incidence is very different from one country to another country, probably because the mechanism is different. But for this disease, it's very reproducible. So we are in front of very stochastic disease, and the results that we obtain are quite reproducible. But it is clear that the immune system is important, and it has been well demonstrated by the stem cell transplantation. And there is something that I don't understand, because if the immune system is so important, and we know that the viability of the immune system is also important, I cannot understand why this disease is has an incidence so reproducible from one country to another country. Have you any idea about this?
Well, excuse me. I think one reason is that it's, for all purposes, chronic phase CML is a single gene disease. I mean, you need just a single lesion to create it, and I think that makes it possible. That may also one of the reasons why the incidence across the world is pretty much identical. So these events apparently happen at roughly the same rate, and at roughly the same rate lead to transformation. I mean, I have a lot of questions why the clinical behavior is so different across patients. Is that, of course, we all think about the same things, like, is it the duration of the disease? Is it the early acquisition of a secondary mutation? Or does it have to do with the state of the cell that gets transformed initially? So in other words, is there something that, so if you look at stem cells, they are, some are fast cyclists, some are very quiescent, some contribute a little bit to hematopoiesis, others contribute a great deal. So you could say there is Volkswagen and there is Porsche. And if you transform a Porsche, you're gonna drive fast no matter what, right? So you bring that. And I think at this point, we don't have, in my mind, probably don't have the tools, although I think the work that Dr. Tiang presented at the CML meeting a couple of months ago is really going into that direction to think about differences at the very beginning. But apparently the hematopoietic system of people is not so different across races and continents and populations.
Okay, we better move on. So thank you very much, Mike, for the great presentation.